The next paper in this session is entitled Investigating Circuits of Conditioned Fear and Avoidance by Greg Quirk, University of Puerto Rico.
Okay, thank you, Ken. And I want to thank USIC and Yuri for inviting us to be part of this ICAN Center. It's great for our lab, but it's great for Puerto Rico, for the whole education aspect and community aspect of the center. We're really glad to be a part of it. In fact, I'm thinking that Michigan is nice, but I'm thinking the next meeting for ICAN could be right here in Puerto Rico. There's a lovely little place right here with a beautiful view. What do you say?
Winter.
NSF, right, and NSF will groan at the cost of it, I'm sure. So what I'm going to talk about, we're not using MEMS. We'd like to use them, and we plan on using them as part of this center. But I want to give you an idea of the questions that we ask about emotion and about fear that we would use this technology. So this is Puerto Rico. This is an island off the coast called Cayo Santiago that is a rhesus macaque research facility where wild monkeys are there, and it's since the 1940s. We do some research there I won't be talking about. So what makes us afraid? It's thought that there are innate fear stimuli like spiders or heights. This is the CN Tower in Toronto. It's terrifying if you've ever walked across something like that. But in a sense, your amygdala is saying you're about to die, but your frontal cortex is saying, they don't kill tourists in this state. Just go ahead. But it's amazingly difficult to do. But most fears are learned through some kind of experience that we've had or we've heard about others having through a process of conditioning. And we can respond to stimuli in different ways. It could be a passive response that we model with freezing in animals, or an active response, this fight or flight, running away from a disaster. And we study both of these types of responses. So the general questions of the lab are what regulates the expression of these fear responses, and how do those fear responses compete with appetitive drives? So the passive fear we study with fear conditioning, for those of you who haven't seen it, we have rats pressing a bar for a food reward. And when this light comes on, that's when they're going to hear a tone for the first time. That's the first presentation of this tone. It means nothing, and they continue eating. Here in the conditioning phase, the tone is on and it will terminate with a shock. Okay, we do that a few times, and then the next day, this is the first time they're going to hear the tone. Okay, that's freezing. The film has not stopped, but you see this guy here is trying to break through his fear to get to the food. There's this competition between these drives, appetitive drives, and fear, which we think is very important for the system. So of course we can record with video methods that the freezing occurs, increases during conditioning, and in extinction we can give the tone without the shock and the freezing dissipates. And then the next day, animals that had extinction training remember their extinction experience. But extinction is not forever. It's well established that if you just wait a passage of time and then play the tone again, it gradually returns or spontaneously recovers. In this case, back to 100% of its original value. So extinction is not erasure, it's some kind of inhibition. And really then the question is, what are the circuits that regulate this fear expression? And of course, the key circuit for the fear conditioning is the amygdala, and it gets this tone and shock inputs from the thalamus and the cortex, and then projects to areas that produce conditioned fear responses. And it's regulated by the prefrontal cortex, which has reciprocal connectivity with the amygdala. And we focus on different parts of the prefrontal cortex, the prelimbic and infralimbic subregions. We can use mucibol to temporarily inactivate, a GABA agonist, to temporarily inactivate these areas. The prelimbic cortex, when you inactivate, you get a reduction in the freezing response. So it's important for the expression of this conditioned fear. But in contrast, the infralimbic cortex when you inactivate it, they express the freezing fine, but they have trouble inhibiting or extinguishing the fear. The next day, they act like they never were extinguished. So they have really opposite effects, like a gas and brake on the expression of the condition fear. And they have slightly different projections to the amygdala as well. The prefrontal cortex targets this basal, lateral part of the basal nucleus, whereas the infralimbic targets this area between the central and the basolateral nucleus that contains these intercalated cells or other types of inhibitory interneurons which inhibit the output cells of the central nucleus. So it's like an off switch for the amygdala that the infralimbic cortex is able to access. So when you record from prelimbic neurons, they show a marked resemblance to the actual conditioned fear response. The freezing is in gray bars, and the firing of an example neuron is in blue. And you see there it expresses as a z-score normalized to the pre-tone activity. You can see it correlates very well with the freezing response. And inputs from the amygdala are critical for this conditioned response. It relies on inputs from the amygdala. So this kind of work has led to these sort of simple circuits that could mediate the expression and the extinction of the fear. The prelimbic driving or exciting basal nucleus and central nucleus to produce fear responses, and the infralimbic giving this feed-forward inhibition through the intercalated cells or through the basal amygdala. So we've been using optogenetic techniques and other techniques to revisit these circuits recently. So the prefrontal cortex does project to the amygdala, but also to other areas. For example, the medial thalamus that we heard about this morning, in this case, the paraventricular nucleus. And it's this area that projects to the central nucleus. So it's an indirect route to the amygdala. And this is anterograde label in the paraventricular thalamus that labels particularly the central nucleus of the amygdala very robustly. So we first started, this is work that we reported last year, we first started by using fluorescently labeled mucimol to turn off this medial thalamic area and ask, does it have any role in the expression of the conditioned fear? So we conditioned animals and then infused at different times after conditioning. We saw a very interesting thing, that if we waited a half an hour or even 6 hours after, this area was not involved, it had no effect. But longer times, a day or multiple days or a month, the freezing was reduced, suggesting that this area is important in the expression at a later time point. So which areas, which subregions might be involved? For that we used CFOS, comparing these two different time points, when it's not and when it is involved. 6 hours after training, you get some slight labeling of neurons in this area. And this CFOS is one indicator of physiological activity in neurons. But 7 days after, you see a robust increase in this paraventricular region, but not in this neighboring mediodorsal area. So counting these cells, indeed, we see this at this late time, 7 days after training, you see this increase here, but not here. Now, the prelimbic area is activated at both time points, and the amygdala is interesting. The basolateral amygdala is activated, as you'd expect, at this short time point, but surprisingly was not activated at this long time point. But the central nucleus was. So suggesting that the circuits may be different at different times. So this more traditional circuit of the prefrontal cortex projecting to the basolateral amygdala may be key for this 6 hour time point. But at 7 days, it may be an indirect projection from the prelimbic to the thalamus and the thalamus to the central nucleus. So we wanted to ask, are these pathways actually necessary for the expression of the fear? For that, we could turn to optogenetics, using either channel rhodopsin or halo rhodopsin to either turn on or turn off neurons or their targets. So we observed in anesthetized animals that applying halo rhodopsin and activating halo rhodopsin with yellow light does indeed turn off the firing rate of the neurons. We can also infuse the halo rhodopsin in the prelimbic cortex, but shine the light in the thalamic target and record from the thalamic cells. And when we do that, we also see inhibition, consistent with an excitatory projection that's just turned off. But in addition, we see excitation. which is consistent with some kind of feed-forward inhibitory circuit. So both occur. This reduction is more prevalent, but you still see this. It was something we have to keep in mind with these sorts of experiments, is that there is often feed-forward inhibition or disinhibition can occur as a result of turning off these neurons. So what about behavior? So here we can infuse the halorhodopsin in these various places at both time points or in the same animal and ask, is there any change in the circuit? And what we saw is that inactivating the soma or the PL neurons reduced freezing at both time points, so consistent with the FOSS data that it's involved at both. But inactivating the projections from prelimbic cortex to the amygdala only reduced freezing at the short time point, but not the long time point. Then if we silence the projections from prelimbic cortex to the thalamus, we get the reverse effect. It only reduces the freezing at the long time point, but not the short, suggesting some kind of switch from this projection to this projection with time. Then we can inactivate the projection from the thalamus to the central nucleus of the amygdala. Again, only reduces freezing at the long time point, 7 days, not the short. And notice the next day, when we present the tone without the laser on, there's a persistent reduction in the freezing, suggesting that activation of this pathway may be important for maintaining the freezing, almost like a reconsolidation blockade of some sort. So the implications of these findings are that the current models we have of regulation are usually based on this 24-hour time point and need some rethinking. And this work is often applied to post-traumatic stress disorder, which are certainly older memories. So this kind of shifting with time could be a factor in either the etiology or the treatment of that disorder. And then the questions we're addressing now are, what are the mechanisms of that shift, and what are the advantages of recruiting this area of PVT? And we're just beginning to get at these issues, and that's what I wanted to talk about. If you look at the prelimbic neurons that project to either target, we can do that by injecting retrograde tracers in the amygdala or the thalamus, and looking at where you get retrograde label. And it's a little hard to see with this lighting, but you can see that it's the deepest layer cell, layer 6 really, that projects to the thalamus, whereas the amygdala receives inputs from the more superficial layers, 2, 3, and 4, 5. So that's fortunate, because it said that different layers project to different targets. So we are exploring the idea that the prelimbic cortex may control this shifting or trigger the shifting by shifting itself, the activity from the superficial neurons to the deep neurons. So the first question we asked is, what are the tone responses of neurons in these two different layer areas, and do they differ with time? So these are examples, single cell examples that we see from prelimbic cells in the superficial versus the deep areas. And indeed, we did see some cells that the superficial cells increased their rate at this hour and have pretty much lost it by 7 days. And the reverse seems to be occur in the deep layers. There's actually an increase with time in the response of these cells to the tone. But we've only looked really at a handful of cells, like 15 cells at this point, but it seems that there may be this shift, this layer-dependent shift, and it also looks like there may be some interaction between the neurons, maybe inhibitory interactions, where you see excitation here but inhibition here. So these are the kinds of things that we want to look at when we can record many more cells. So this shows individual cell examples from these two different layer categories. Indeed, there's a pronounced effect here, the superficial cells at 2 hours, and the deep cells, there are some cells that increase their responses with time, some don't, some seem to get inhibited with time. If you average across cells in these two different layer categories, it looks like there's nothing happening at 7 days at all, but again, I think that is an inappropriate average, but this layer-based ID really may not be sufficient. We really need to know what are the targets of the cells and be able to identify cells based on their targets. So that's where ICAN comes in. If we can have probes that will record from multiple cells, and using this LED technology, we can then cross that with an intersectional strategy where the channel rhodopsin, we have HSF-Cre, which will retrogradely transport, intersect with channel rhodopsin diode. So you basically only the cells that project to the PVT will express the channel rhodopsin. The same can be done in the amygdala with a red shifted channel, so that we could basically identify or tag neurons and know what the target is at the time we're recording. If we can record a lot of cells, we can also talk about the interactions between the different layers and get cross correlations and timing effects on these areas. So this is really the project in this grant that we want to do, one of the projects. Now, what are the advantages of recruiting PVT into the FEAR circuit? One may be, as I've already mentioned, maintenance of the FEAR memory through some kind of reconsolidation process that's occurring. And there's evidence from that from Bo Lee's lab that it may involve BDNF. But if you look at the connections, PVT is uniquely connected. It receives inputs from all of these different areas in the midbrain, hippocampus, amygdala, and projects back to many of these areas, and also projects to the nucleus accumbens. So it may be uniquely situated in terms of the anatomy. So other possibilities is that the paraventricular thalamus could coordinate fear memories with other responses, such as arousal, chronic stress, or reward and food seeking. And this is an area that we're particularly interested in, we're looking at. And we've developed a task where the animal learns that the bar will only produce food when this light is on. So it's cue-induced. So he just waits for the light to turn on and then presses to get rewarded. So in this case, we can train animals this way, and they learn to press, in a few days, they learn to press only when the light is on and not when the light is off. Then we give another day with the food available as before, or we turn off the feeder and just omit the food that the animal is expecting. So we first started with fluorescent mucimol to inactivate this paraventricular area. And we observed that we inactivate just prior to this test on day 4, and it had no effect when the food was available. But when the food is omitted, it actually increased the firing rate, increased the press rate when we inactivated this area, suggesting that the paraventricular thalamus suppresses pressing, but only under these conditions where the expected the reward was omitted. So we then turn to activating the paraventricular thalamus, and would that inhibit the food seeking? So here we switch to channel rhodopsin expressed here in the paraventricular thalamus, and when we activate or stimulate these neurons, food seeking is reduced both when it's available and when it's omitted. So this seems to be a switch to turn off that food seeking. And I think there's a video here showing that. So here, the laser's off, it just gets the light, and of course, both animals will press for food. But here, the laser will turn on just as the light turns on in both animals, but only this animal is expressing channel rhodopsin. So it's hard to say, is he not hungry, or he doesn't really know that the bar produces food, or it doesn't have an effect on spontaneous pressing without the light. So it's not, we don't think it reduces the hunger of the animal. Okay, so the next question we ask is, what about specific projections from the paraventricular thalamus to the nucleus accumbens? So here we can infuse the virus in the paraventricular thalamus and shine the light in the shell of the nucleus accumbens, which we're seeing here, which receives the main projections from paraventricular thalamus. So we asked if this would have similar effects to the mucimol. So no effect when the food was available, so we shine the yellow light right here, but when the food was omitted, again, the same increase in pressing that's occurred there. So we think this is It's not aversive, but it is a negative consequence situation. The animal is used to this one-to-one. It always gets a pellet with every press, and then there's no pellet available. So what we think is happening is that under conditions where an animal may have had a bad experience or a near miss with a predator in a given situation, and it returns to that area and there's some stimulus that is potentially dangerous, the prelimbic cortex can activate the paraventricular thalamus. And this can, through projections to the central nucleus, turn on fear responses. And through projections to the nucleus accumbens can turn off food seeking, which is, again, part of the fear response. And these can be coordinated in the paraventricular thalamus, but we don't exactly know to what extent that may be. And again, this is where the silicon probes would be key in helping us to record between these cells. Are they the same neurons or different cells? Do they interact, et cetera? Okay, these are the people who did this work. This is the postdoc Fabrizio Del Monte from Brazil, who brought optogenetics to Puerto Rico and did a great job. And this is a MD PhD student, Kelvin Quinones Larquente, who did all the unit recording. Kelvin is here at the meeting. And this is a super undergrad that's been working together with Fabrizio for a couple of years. And she is actually part of, in the ICAN summer program, and right now she's in Ileana's lab in Hamburg. And here's the whole group on the roof of our building, and I'd like to acknowledge the funders and also NSF for this program. Thanks very much.
Lamborghini, silicon fabrication, there are minimum volumes of devices that have to be sold or you can't afford them. You know, the first probe costs $1 million, and the next 1,000 cost 100,000.